Hey, boys and girls, weather report for December 27th, 2022. I know it's not December, but it sure as fuck feels that way. What is wrong with the weather?
It feels nothing like December where I'm at. It's 73 degrees, partly cloudy.
Oh, get out of here, you fucking Texans.
Remember, in a couple months, I'll be dreaming of December.
Yeah, it's like, what do we got here today? Jesus, it's 46. It's supposed to be like in the 60s now, you know? It's like, Jesus, what is up with this?
Well, that was the weather report, people.
That was the weather report. Anyway, April 27th, 2022. Yeah, 26,044 days old.
I have a correction I need to make, Speed.
Oh, yeah, go ahead.
Well, yes, the other day, I guess it was yesterday, did we do the 8 o'clock thing? The morning Joe, I guess. I don't know. But I kept mentioning that it was Judah and Dan together were the only two tribes of the Southern Kingdom. Dan is turns out is in the Northern Kingdom. I for some reason I just in my mind replaced the tribe of Benjamin with Dan, probably because I've been thinking about Dan from all the reading. So it's Judah and Benjamin were the Southern Kingdom, and Benjamin was just a really tiny area, a little tiny tribe or whatever, and then Dan was in the far north of the Northern Kingdom. So I just wanted to issue that correction, and also Andrew Cumming gets a prize for noticing that I'm wearing a new hat today.
Oh, nice new hat.
I forget the name of the type of fedora it is, but it does have a feather in it. Stick a feather in his cap. But anyway, and as a prize, he's going to get it's something new from a brand that we've all known for years. He's going to get some new Wonder Bread instant toast. That's right. We're going to send Andrew instant toast.
Instant toast
from Wonder Bread.
You pull it out.
At least actually we'll send him a coupon for it. You never really get sent food stuffs, but that's going to be great. Congratulations, Andrew, and thank you for noticing. It makes me feel semi-alive.
So I'm looking at your diagram. I don't see 650 BC. I see you lie. You're lying to me.
I didn't lie. It's just a big page and it's confusing.
I know. I'm so confused right now. Anyway, yeah. So what the hell did I use for a thumbnail today? I used what is it?
A fish?
Pterospidomorphy.
That's why you brought up Just a Walking Fish. You were looking at pictures of fish.
Yeah. So
Walker, if you're out there, I raise my glass to you, and I say we miss you, buddy. Wish you could come and join us.
So, I got to tell you an embarrassing truth about it. Evolution. When it comes to conodonts and cyclostomes and pterospidomorphs, we're still fighting about about how these things are connected, basically, because the fossil evidence is there, but it's a little hard to figure out because a lot of these things, we're just developing the whole bony thing, and so a lot of them are, so it's kind of obscure and there's a little bit of fighting going on, but we've got, we keep coming up with new ideas. And if you take a university course in paleontology, the first thing they will tell you is that if you buy a textbook today, it'll be obsolete next week because we're reclassifying things, all that stuff. But these guys are kind of cool. They have big bony armor plates and their vertebrates, their chordates. I'm not sure if they have a well-developed vertebral column or not. But anyway, looking into ancient fish, there's a lot of fishies.
Are you looking at something that you could share?
Yeah, I could.
Okay, because
I don't think I want.
That kind of helps.
I don't know if I want to share.
Well, we'll have to put that on your report.
Yeah.
That you don't like to share with others, and that would look pretty bad.
I'm going to share today. Looking at this guy. So this is a Wikipedia article. An extinct class of early jawless fish, they have long been regarded as closely related or even ancestral to jawed vertebrates, but they don't have jaws. But they got big bony heads. But the few characteristics they share with the latter are now considered basal traits for all vertebrates. So I don't know. We're still trying to figure this stuff out. They're characterized by dermal head armor having large median ventral and dorsal plates or shields. And they're kind of pretty. At least the artist's rendition is.
I know. They're just making it up.
Yeah. So I sent that Andrew guy a link.
Yes, Andrew guy.
Andrew guy, who seems to know everything.
He seems to know a lot about this. He might be a great contributor to the weather report today.
Yeah, so I'm just a very early student of vertebrate paleontology, and what I need to do in my brain is I have to put together a tree, and I have to place everything in the tree. And to do that, if you look at an article, you'll find several different trees that have very slight differences between them. So what you do is you keep looking at it over and over again and putting it in your head, and eventually you come to a whole understanding of where everything fits. And that's kind of where I'm at right now, and I'm just trying to do that. And then, of course, you got to place it in time. But now, okay, for our biblical studies, so the thing we got, Dan. So Dan was a dude who had many sheep, and he had a kingdom. Is that what Dan did?
No, it's just a tribe. It's
the tribe of Dan.
Yeah, the tribe is named either after one of the sons of Jacob or somebody related to a son of Jacob. So,
but was the tribe named after him because he had many sheep?
Why are you focused on the number of sheep?
Why? What is my okay? Okay, I'm not going to answer that. I plead the fifth.
Yeah, the guy's name was Dan.
I'm taking the fifth on that.
And it's not Dan because he had a lot of sheep, unless I'm just forgetting a story. But yeah, the guy's name was Dan. They named a tribe after him, and that's how they divided up the territory, was by the 12 sons of Jacob, or as he came to be known, Israel.
But that's a biblical story. So
do
we have facts about Dan?
We have no facts about Dan that I'm aware of. We have the city Dan, or the tell Dan, where they've discovered lots of things that are archaeologically significant. But
the sheep?
Yeah, it's nothing but sheep up there.
Okay. So they've
sheep as far as the eye can see.
But did they know that it was the tribe of Dan where they found these archaeologically significant things? They don't know that.
Well, technically, I'm not quite sure. It's Tel Dan, so the area was named Dan, but and of course, you know what a tell is? It's just a mound where a city used to be, or a community used to be, and over the years, it basically got leveled and built up on, and leveled and built upon again. And so a site in Israel and other places, I'm sure, are just going to be mounds, raised areas that contain all the rubble from before.
So that's called a tell. And so there's a tell Dan.
Tell Dan, yeah.
Tell Dan. Do they? So we know where that is geographically now, right?
Yes. It's been excavated for quite a number of years. They found quite a few significant things. If you what? I'm trying to remember what documentary that I watched. I think it's The Bible on Earth. There's a video series based on that, and I think Israel Finkelstein spent a lot of time there. Don't quote me on that, but that's the impression I get. That's what happens when you start binge watching all sorts of YouTube videos on similar subjects. They all run together.
They all run together. Yes. So
speaking of YouTube videos, you made one yesterday?
We did.
Or live with you?
Yesterday morning.
And I noticed that you guys, or Speed, you wrote down year 0.
Yeah.
In your one, there is no year 0. There's like year 1 or -1.
Oh. Well,
he's right about that. Technically,
I'm a mathematician, so
just to let you know.
Yeah. So I would put 0 right between -1 and 0 then.
Now, did Andrew give a reason why he's not in here yet?
Yes, my right
eye. He's asking you
really
interesting questions, but he's not here with us. Like we're not like in anything. We're just in a conceptual space known as here.
Yeah. Yep. So, did you notice the little numbers by each node of most phylogenies yet? Did I? I think I have. And those are the number of, well, it depends on the phylogeny. I don't know. Why don't you come in here, Andrew? What the hell's wrong with you? Yes, my right eye looks red because of the way that the screen windows are showing on my eyes. Like I have really deep eyes.
Deep set, you mean?
Huh?
Deep set eyes?
Deep set eyes. Well, there's wrinkles there, you know. They're getting droopy. Getting old, getting ancient. Ancient in a paleological sense. Oh, let's see. So, I don't know. What are we talking about? Oh, so we got a really complicated job ahead of us. So we got a timeline, right? So we have historical things that we're pretty sure of, and they have a date. And they also, every one of those events has a location on the earth and a date.
Mm-hmm.
So there's a geography to it, right?
Right.
And then overlaid on top of all of that is what the Bible thinks. So the biblical
narrative.
Right. And so then you got to compare the two and find out if they make any damn sense at all. And that is a big task.
Mm-hmm.
And what I'm, and what we were talking about backstage is doing a little piece of creating these little databases or little data files in comma separated value format to at least get started with some of that so that we, and then, type, excuse me. Type would be, that type column, the type of event. Like, was it a war? Was it, whatever, somebody born or whatever? Could also be biblical narrative, and there could be a super type, right? So according to the biblical narrative and according to what we understand from history and archaeology.
Mm-hmm.
So that would be like a fourth column. So that would be a pretty cool thing to do.
Yeah, just to help keep up with it as we try to cram actual facts into our brain.
Yeah,
and compare them to non-actual facts.
Somebody's threatening to come in.
They are.
Yeah.
Well, I wish they'd hurry up and put the pedal to the metal. I don't know.
She'd hurry up and come in.
Yeah, Andrew.
Yeah.
He says he can't stay a while.
Why? I want to know why.
Yeah, probably has to like do things like
Oh
daily
my God.
activities for life.
So Danny had a live stream last night. I should have stopped in. Cell, Rebecca, Jason. Oh, I should have stopped in because Cell was there. See if Cell disappeared. That would be interesting.
That would be interesting.
It would be an interesting experiment. See if I can brand myself as Cell Be Gone.
Now from Ronco, Sal be gone.
Cell Be Gone. I'm choking. Kind of waiting for Andrew to come straighten us out.
Waiting for Andrew.
Waiting for Andrew.
That's kind of an update to Waiting for Godot.
There he is.
We have a rational empiricist aboard.
So what are the little numbers? Are those number of differences or mutations, or what are the numbers?
Let me show you. Hold on.
I've seen it a couple different ways.
Yeah, there's a couple different ways of showing it. I'll show you the specific one I'm talking about. All right. If you can, please share my screen.
Let's see.
He's a very polite gentleman.
He is. We're gonna do it together. Oh, it's popping up.
Okay, can you see it?
Yeah.
Can you see my mouse?
Yep.
Okay. So these are the little numbers I'm referring to. So one of the most common ways of measuring the statistical support of different phylogenies is this method called bootstrapping. Now, it's not the only method, but it's kind of one of the simpler ones and one of the ones that I understand more. So, so basically, what you're doing is you're taking the different branches and kind of taking the character data that you're using to construct the phylogeny, and you're kind of varying it in small ways over a bunch of different times and seeing how many different times the same phylogeny, or in this case, the same branch points, nodes, pop out. So if, for example, you do this bootstrapping and repeat it 100 times, for example, just using a hypothetical example here, then the 95, or probably more a decimal, would indicate how many times out of those many repetitions you got that particular node from varying the data. So generally, the higher the number beyond a certain point, and the certain point, as far as I can tell, isn't agreed upon, at least not rigidly agreed upon, but basically the higher the number, the more statistically robust, I guess you could say, the particular node is, and whether that means it's true or actual per se is kind of up for philosophical grabs, but yeah, that's what I was referring to.
Okay, so if we if the characteristics we were displaying were so we're comparing two genes, right?
Okay.
Okay, how would that hash out? How would you do multiple trials? If we compare more than one gene, like, okay, we're comparing genes A between the two, and then we're comparing genes B and then gene C. So would that be 3 different trials?
As far as I understand, at least for the methods that I understand the most, you generally do a phylogeny, right? I guess you could say, I believe it's parsimony for the most part that does this. But basically, you generate a phylogeny for each tree, or sorry, for each gene in this case. So, for example, what's the simplest way you would get to that gene? The more specifically the fewest number of evolutionary changes that would generate the changes you see in that gene. Okay, so re-ask your question again. Sorry, I kind of got.
Okay, so I'm trying to relate the number to some actual data. So if we were to actually run one of these, what would we start with? We have two genes. We have A and B. Okay, and we have three genes: A, B, and C. Okay.
Okay.
So A, B, and C, and we have two different species, or a bunch of species or whatever, and we compare genes A and B, and we come up with the number of mutations between, or A and A, across two species, and we come up with the number of mutations, right?
Well, if you're talking about the number of mutations, like in total, right?
Yep.
That's it's kind of a different method than parsimony methods. For example, it's kind of more so a.
Let me ask you another way. So
okay.
when you're talking about this, okay, when you came that little
you can compare the total number of differences, or you can compare the specific nucleotide at each site along the gene. There's kind of a different method there between them.
Depending on whether you account for alignment or whatever. Is that what you're talking about?
Well, you can. So there are like phonetic methods, or I should say distance methods. The ones that come to mind are neighbor joining and least squares, I believe, and UPGMA. I hope I got that acronym right. Basically, you're kind of aligning the sequences, and you're doing a summation of all the total differences between them, and the most similar, according to this, are going to be more closely related. Whereas in parsimony, you're taking basically each character for what it is and finding the way that would lead to that character in the simplest way possible. So the difference is kind of taking everything at once versus taking each character by it is and seeing how it could have gotten that way in the most likely way possible. Okay. If that makes sense. So it kind of depends.
In the diagram you showed, what is the data that you're working from? What are these characters? What is the data? What does it look like?
It can depend. It can be morphological data, molecular data.
Okay, so we had morphological data. So where would you learn all about?
Yes, correct, Nestle. That's what I was trying to say right there. Thanks.
Okay, so I'm trying to drill a little bit deeper into this so that we can explain it better. Exactly how they come up with these trees, right? So morphological data is kind of a really deep sort of subject, right? If you're looking at, you could look at like has a basically it's a vertebrate, or it has a cranium, craniata or whatever. We can look at those things, but morphology is kind of difficult. Is there any way that we could explain it, the data? Explain it with a different data set. I like genes, and nucleotides. Because you have strings, right? So,
right, yeah.
Okay, so if we ran our phylogeny based on gene A across 20 different species, and we come up with a phylogeny, and then we ran it again using gene B, basically comparing the two, and then we ran it again using gene C, okay, and they all came out the same, would that give us that would give us 100%, right?
If they all came out the same for each gene?
Yeah.
Yeah, I mean, probably, but that's probably not going to actually happen, most likely.
Okay. Yeah, it doesn't matter. Okay, but if they came out, two came out, so two of them came out and one didn't, then we would have a 66, right? So what I'm getting at is that number a percentage of the number of trials that came out with that same phylogeny, with that same structure.
Okay.
I don't know what the number
Speed,
means.
real quick, just to interject here. Looks like this Grr person. Have you heard of him?
No.
Okay. has
know?
has a live stream at 5:15.
What?
Yeah.
5:15 our time?
All right. Oh yes, because I looked at it on my Chrome and everything, and the time it's given me is 5:15. So we might want to shoot it in the head in 45 minutes, or we might just want to do a whole hour or two of fuck you Gurmania.
Well, if Gur is going to be on YouTube, I don't want to be on YouTube at the same.
Oh, you don't want to be on the
I
same
don't want to be in
platform
the
as
same
him?
platform as
Yeah.
him at the same time. So I
Maybe.
definitely will get off before.
Yeah.
Yeah. So,
But then, he never invites me to come on or anything.
you know, it kind of begs the question of why the son of a bitch is starting a stream on Wednesday when he knows that I have the weather report, right? You know. But
don't know.
anyway,
I don't know.
we'll get into that and many more things on his stream
Yeah.
later on. Okay.
Yeah, just get on there and bitch at him about it all.
Yeah.
Between every other comment of every other person.
Anyway, Andrew, I'll quit asking questions about that, and I'll go figure it out myself, and I'll tell you what I'm trying to figure out. Okay.
Okay. Sorry, I couldn't help you much there.
Yeah.
I don't think I quite understand what you're asking for.
Well, basically, you've got a number there, right? It represents some quality of something statistically, right?
Yeah, it measures how from varying the initial data set in specific ways or just random ways, how much you recover that particular node of the phylogeny out of the total number of repeated variations. So if I like I said, do a phylogeny and I then vary the data set and repeat that 100 different times, if I get the same node 90 times, and 90 is going to appear at that node on the consensus phylogeny.
What if you do it 60 times and it repeats 50 times?
Then it would be a different number.
Would it be?
It doesn't have to be like 100. I'm just saying that's kind of the at least 100, maybe 1,000. You kind of want to do it as many times as possible.
Okay, so that number may end up being 365. It's not a percentage.
I don't think it's a percentage as far as I can tell.
Okay, I'll look into it. And then when you say, see, the problem I have when you say varying a data set in different ways, that leaves two questions. Number one, what is the data set, and how are you varying it?
Well, I mean, you can take an alignment sequence, for example, and if you're comparing the same gene between many different organisms, you can kind of switch some of the characters, in this case, the nucleotides, along the same column as you're aligning it. And that way you can kind of just, like I said, vary the data. Basically, you can, it's really hard to explain this without a picture, but
Yeah.
Does that kind of make sense? Like you're taking this alignment sequence and you're randomly altering the specific nucleotides, let's say in this case, at each site along the gene.
How can you do
in
that?
these different species?
How can you randomly alter the nucleotides?
That's beyond what I can tell you.
Okay. Because, yeah. So if I take three different genes from three different, or one gene from three different species, I end up with one alignment with three different, basically three different nucleotide sequences. And then I determine how many differences I have between those two after I align them, which accounts for gaps and deletions and insertions, right? So we have a rough alignment. So I'm just wondering about this variation thing, but I'm putting you on the spot, and I probably shouldn't.
Yeah. Well, like I said, you should probably keep in mind, like I said, that not all phylogenetic methods involve taking a census of all the total number of nucleotide differences. So
right.
That's also another thing to keep in mind.
Well, what are the other ones? Like morphology? Are you talking about like morphology and the difference between morphology and genetics?
No, like I mean, like even with molecular data, oftentimes, like the simplest, probably fastest way to do it is the way you put, which is to just in a complex way count the number of differences between the data sets, the genes in this case, between the different species.
Right.
That's one way to do it. Another way to do it is different kinds of what's called parsimony methods. And a further way of doing it involves something called maximum likelihood, where basically you have a specific model of how each of the characters, in this case the nucleotides, could evolve over time. And based on that model, you're trying to find the likelihood that you would get the data you would observe given the tree in that particular probabilistic model of evolution. So that's kind of the basic idea behind it. It's some rigorous mathematics behind it that I don't know how to get into, but
Okay.
and then the kind of a breadder, I guess you could say, of maximum likelihood methods that are somewhat more computationally less intensive are Bayesian methods that are kind of the reverse. They involve prior probabilities. You'd have to get into statistics, of course, about that. And basically, instead of looking at how likely you were to get the data given a tree, you're trying to see how likely you are to get a tree given the data and some prior knowledge about the data. So it still involves like a model of molecular evolution in this case, but it's kind of the reverse.
Yeah. Yeah, I'd like a good, I don't know. I've got a lot of textbooks on this stuff, but I'd like one that actually had some, where you actually worked some real problems, did some real problems with real data.
Well, yeah, you can find like examples of people working it out with parsimony on like YouTube, but I don't know if you'd have much luck finding it with more complex methods.
Yeah. Okay.
At least that's what I've tried.
But anyway, the number is like a reliability number, and I guess my question about the number still stands: is it 0 to 100, or are we talking about I don't know, something else?
I guess it just depends on how you're phrasing the question, or how you're phrasing the bootstrapping.
Yeah, can you link that example to me that you had a picture of?
Sure. It was.
Private chat.
I don't think it was a really rigorous site. I just pulled up a random image example, but.
And then we're going to switch off to picking on Garte and Bread of Life.
Yeah, I was going to say that Top Dog was took the initiative and provided us a timestamp of when he showed up on.
Likelihood judges each possible tree. The number of possible trees is insanely high for just a few samples. Gives a probability value for each node, a total p-value for the tree. Shit.
Oh, yeah, that's not right, Nestle.
Yeah.
All right, there you go. I put the link in the private chat.
I really appreciate Nestle's little notes there.
Yeah, he's keeping me on track here.
Mm-hmm.
Yep. Good.
And I appreciate that because I can stumble.
That's you. You want trouble. Oh, I'm sorry. I'm talking to the dogs.
You are talking to the dogs.
Yeah, or just real quick to comment on what one thing he said there. Like he is correct that if off the top of my head, if you go into say 20 species or so or more, you're talking about more possible trees. Just to give you an idea here, more potential different ways of connecting the relationships between these different species. So
then there are particles in the universe.
Yeah, then there are particles in the observable universe. So it can get really tedious.
Evolution isn't real.
Yeah.
Yeah.
Yeah. I guess before we look at this video, I want to mention Lee Cronin and Sarah Walker are working on something called assembly theory. And Sean Mack kind of led me to it. Lex, we linked it in I think weather report on Monday, but Lex Friedman interviewed these two, and they were talking in depth about this assembly theory. And what they're coming up with assembly theory is kind of it's a measure of whether something was created by life or whether it was just something simpler and something they wouldn't call life. So they're coming to the point where, and I know the intelligent designers are going to use this and claim they're showing that a mind had to be involved. But that is not what Lee Cronin and Sarah Walker are doing. They're showing how life has to be involved. In other words, there has to be a past, like a memory. They talk about a memory in a system. You know, that DNA is a memory. The cell membrane is a memory. The cell contents are memory. And it's really an interesting theory. And we can brace ourselves because we are going to get an onslaught of creationist misinterpretations of this theory. And I think they're working on publishing their paper. They're trying to get it through the editors right now. And
not only that, there'll be what we used to call New Age interpretations of it.
Oh, those two. Yeah.
Like, Shirley MacLaine used to claim, as other New Agers did, that water has memory.
Yeah. Shirley MacLaine and Richard Gere are where I got the term Hollywood Buddhist from.
Yeah.
Yeah. They're definitely Hollywood Buddhists, the woo and the crystals. But anyway, I want to look a little further into it, and I'm going to try to reach out to Lee Cronin and maybe get a look at that paper before they publish it, hopefully. And it's a really exciting field. And what they're doing is they're creating graphs. So they take basically a molecule, and if you find one of these molecules, they're creating a graph about how you may create that molecule. And he uses the example abracadabra as a string. And abracadabra has abra in it more than once. So it's a shorter path to create that than it would be to create a random set, with the same number of letters, because you can reuse abra, right? So they're showing how you can reuse components and create a molecule, and they develop a graph. So they have a graph space. There's more than one way to create this molecule. And so what they find is the shortest possible space to create that molecule. And then if you find millions or billions or trillions of those molecules laying around and they have a very long development, they have too many steps in them, right? Then you know that there is life. So if we found one of these molecules in outer space, we would know that it was created by a planet that had life on it. And so he's looking for the signature of life itself and basically what life is. And it has nothing to do with intelligence. It has to do with combinatorial. It's basically combinatorics. And he's got the math to back it up. And not only that, but he said it's experimentally verifiable. So it's something that we can actually measure and come up with good statistical results on it. And it's pretty exciting. Anyway.
Hey, Speed, real quick.
Yeah.
So I just posted in the private chat a link to a particular article by one of UC Berkeley's website about bootstrapping and some other things related to that. You can also find if you want to look at that, or for the audience's sake, or if you want to put it in the description.
Okay, I'll throw it in the description.
Because I think it's a pretty good website, but
okay,
you can judge me on that.
It's Berkeley. Of course it is.
Yeah.
What else do you want?
It gets the basics across pretty well, I think.
Yeah. Okay. Let's see. Bootstrapping.
So that's all I have.
Okay. Yeah, we're kind of dissociated today. So I was looking at conodonts. I'm looking at early fishes like cyclostomes.
Yeah.
because they have extremely simple. Well, in fact, cyclostomes, their salinity is the same as the ocean, so they have very, very primitive kidneys, like the most primitive kind of kidneys you can have. They don't do a lot of adjustments for sodium ions and things like that. So that's why I'm looking at cyclostomes, and they're interesting critters. And we have 2 of them that are alive today. I think about 100 species, but there's 2 branches: the hagfish and the lampreys.
Yep.
And they're pretty cool. And then, of course, you look at the conodonts and all the other crazy things that basically went extinct, that we're trying to figure out.
Yeah. And yeah.
But I'm very interested in them because of the very primitive digestive systems and kidneys that they have. And we still have some that are cooking today. They haven't evolved a more complex system like we have.
Well, or less derived in respect to other vertebrates.
Less derived, right? And so they found some sort of a niche, and that niche happens to be a certain salinity of water and a certain temperature off the Atlantic and off the off the north end of Europe. Basically, that's one of the few places that we find them now, and they're probably going to go extinct because they're whatever. They might not. They might stick around.
Well, let's hope not.
And it also leads us to a discussion about why some things appear to be living fossils, and they appear to be living fossils. They show us very, very early primitive beginnings that we probably took off from. And so I've been looking into the biochemical pathways that create them, that basically the embryogenesis of these animals, and how these pathways are very similar in the early phases of our embryogenesis.
Well, personally, I don't really like the term primitive characteristics, because when you're looking at humans or any other living species for that matter, you're going to see a combination of ancestral and more derived characteristics.
Right.
So even for so lampreys and other living fossils, quote unquote, aren't unique in that regard.
Okay,
Just
but what you want to
there.
consider is that, okay, when you go from the zygote to a certain stage of development, okay, we share the same pathways with them. So for the first few weeks of our embryogenesis, of our development, we share the same pathways as they do, and they stop, and our kidneys go on to further develop. Our kidneys go on to, and you don't want to say further, and I don't want to say more positive, because here's the problem. Because we have these more complex systems that have developed, we cannot regrow our kidneys, whereas some of these primitive or less derived animals can, because they have stopped at a very early stage in embryogenesis, and they have those stem cells available so they can regenerate them, whereas we're too far down the pathway. So as you get closer and closer to mammals and higher animals, you have a kind of animal that is less capable of regenerating certain parts of its body than some of these other animals. And I keep, instead of primitive, what could we say? Simpler.
Like in terms of the traits themselves.
Yeah.
Well, the technical term for it is plesiomorphic.
That's not good enough.
That's not good enough. Well, I mean, like the thing with, if you're going to call something primitive or derived or like basal, it again, it depends on the clade you're specifically talking about, because what's derived or primitive, quote unquote, for one clade is actually not so much for another clade. So you have to add that with respect to whatever you're talking about here.
Yeah, we don't want to use those terms at all. What we want to talk about is Let me see if I can get this to work. And it's not working.
Well, I heard that.
I'm just saying those are the technical terms for it. If you want to use them, sure, but I'm just saying those terms do exist.
I heard that we can regrow our liver a little bit.
Oh yeah.
Yeah, we can. Yeah, that's one of the things that we can. But unfortunately, there aren't too many things like that.
It's our environment that depends on what we need and for our genes to do their job.
Okay, let me try to explain what I'm talking about, Andrew, so we can figure out whether or not.
I also see Nestle posted something in the side chat.
Nestle has a link he wants us to look at.
Actually, it looks kind of interesting how a platypus would describe a human. I am intrigued.
Was that the Sandwalk link?
Yeah.
Sandwalk link.
And do you want to send a link to Brainbug, or can I just put it in the side chat?
Go ahead. Yeah, go put it in the side chat. It doesn't matter.
It doesn't matter anymore.
It doesn't matter anymore. Okay. So when you start out as a zygote, right?
Yep.
Here's my zygote, Z.
Back in the day when you were just a zygote.
Okay. It basically starts developing and creates, 2 cells, 4 cells, 8 cells, that sort of thing. But it's still, the same tissue type. Okay. But then it branches off into 2 different tissue types. So we have tissue type A and B. Okay. And then this one will branch off again into another tissue type and more tissue types. And we go up a number of different levels. Okay. So
the stem cells are the first cells?
What I'm saying is that the cyclostomes, what did you say, Top Dog?
Are the stem cells the first cells that grow?
Well, no, there can be stem cells at any stage, but that's
okay.
Yeah. There's things called, at the very beginning, when the embryo is fertilized or when the egg is fertilized, right? You have something called a multipotent cell type, right? It's a kind of stem cell, so it's called multipotent, and that will create the umbilical cord for us, the placenta, the egg yolk, a bunch of other things. Okay, but then part of it can differentiate into something called a pluripotent cell, which can generate any part of the organism that is being created. So that's pluripotent, right? Or no?
Are there any omnipotent cells?
Excuse me. I blew it. I'm sorry, guys. These terms are a bit totipotent. Okay. So totipotent. Okay. Now these go off, and some of these things can be called multipotent, and those are stem cells. And that means that they can't create any cell in your body, but they can create all kinds of liver cells or all kinds of kidney cells. So those are multipotent. So we end up with totipotent, which can create placentas, umbilical cords, parts of, all kinds of weird shit that's outside of the organism. And then we have pluripotent, which is the basic first cells that start out creating you in embryogenesis. Okay. So anyway, that might have been a bit of a diversion.
Hello, Brainbug.
Hi, boys. How's it going?
Hey.
Oh, great. Nice to have you
Hey,
here with us.
Hello.
Thanks for popping in.
So what I'm saying is that these organisms become more and more complex because they get further up this tree, right? So there is a direction toward complexity, but it's not a direction toward better. It's not necessarily.
I'd have to quibble about that there, though, because there isn't really an up the tree per se, except in the time direction.
Well, it's temporal and geographic, but yeah, it's
like you're moving. All the time direction is moving from the root to the tips of the branches. You're not really moving upward in the way you're showcasing here.
Okay, it's definitely up the tree because the tree has a base. Because you're starting out right here.
Yes, but the base is a root. But yeah, go ahead.
If you think about the gradient as the up down, what is that? The y-axis, the up down axis, as time. When you look at a tree like this, then that kind of puts it in perspective. That's the temporal gradient, and then you whatever the the other directions can be a geographic gradient or a temperature gradient, something like that.
Right. Yeah. So we're definitely going up a tree because these are all, really complicated trees. Okay. And that's how we're conceiving of this, right? This further development into, from basically the pluripotent cells into all of the different subtypes of cells that you have. Okay. So we're going up a tree, and we're going up in complexity because more things happen out here at this end. Okay. Now, they're not necessarily better because these organisms tend to be a little, they could die in many more ways than these simpler organisms. Okay, but there are some species that stop right here. They stop real early in that tree, and they have developed a complete organism that is perfectly viable and reproduces for 300 million years. Okay. So there's nothing wrong with that species. There's nothing more advanced with the guys that go up here. But you have to look at it as a tree when you're looking at embryonic stem cells and how they divide, because you can't get you have to be here in order to get to here, right? On my diagram. So you have to start here before you're going to get up into these, into these more refined cell types. Okay. That's what I mean by upward. Okay. I'm not saying that it's better or it's, less primitive or anything like that.
Yeah, you're talking about the upward in terms of the branch points, right?
Right. So, what happened is we started out with a certain genome, and then we had all of these duplications, kept duplicating over and over again. And we had viruses that popped in, and they kept creating more and more branch points in the organisms, so that they developed more complicated organs, more like the mammalian kidney is much more complicated than the fish's kidney, which is much more complicated than the arthropods' kidneys. So we have to look at it that way. We have to understand that, okay, that there are more, there were basically, how do you describe it? It's like, it gets finer and finer grained as you end up out on these branch points, because you can end up with 4 different tissue types instead of just one tissue type back here, right? That sort of thing. So that's what we're looking at. Now, cyclostomes stop back here, and I think if they get damaged, they can regenerate a lot of their basically the system that works as their kidneys, because they're much simpler. Whereas we can't, because we have lost these intermediate steps in embryogenesis. Does that make sense?
I guess so.
Okay. So anyway, that's what I mean by more complicated. So every animal ends up with this specific tree based on its genome and based on its development. And then among animals, we see these clusters, of types or species or families or groups that will have very, very similar morphologies to these developmental trees. But all of evolution needs to be understood in terms of embryogenesis, is the claim that I will make. Okay. And
Okay.
if you understand that fully, then you can understand why we have cyclostomes today and why they didn't go any further, and become more complicated for, in 300 million years, even though they changed over all that time. But okay. That's kind of what I'm saying. So, but I want to talk more about that. What do you think of that idea, Andrew? And
about what? Sorry.
Yeah, basically the idea of approaching
about the complexity, right?
Yeah, complexity, and look at these trees. So we I mean, admittedly, mammals are more complex than some arthropods in their development.
Yeah, I guess you could see it that way, but there are other ways you could look at it where they're just as complex as us. So it's kind of subjective what you want to call more and less complex here.
Yeah, maybe. But okay, so if we have like 225, I think, tissue types in the human body. Okay. So if you have a beetle and it has 180 tissue types, which one has more tissue types?
The mammal.
Trick question. No, there's no trick. Yeah. So basically you're looking at a smaller tree. So you're definitely looking at a smaller tree for certain animals. They will end up with less tissue types. And to get to these more tissue types, we have to have more branches out here. So we have to have differentiated more.
Well, the other difficulty with your analogy here is the fact that traits can also be lost over evolutionary time, even during embryological development. So it isn't a ladder-like thing you're showing here.
Okay, so yeah, you're having a problem with it because of a way of thinking that we're coming at it from two different points of view.
Well, I think he's just talking about selection, though. It's just that being a pressure that's going to remove some of this, right?
Yeah, at a population level. So basically, over time, it's possible that this entire branch of tissue development could be removed. That this gene mutates, and yet the organism is still viable. So we would lose something, but that just complexifies this. So.
And it also can be clipped, of course. Like the idea.
But if you take any,
it'd still keep growing. Yep.
If you take any organism, okay, there are going to be a finite number of tissue types in the adult organism, in the adult reproductive organism. And if there's a finite number of tissue types, then each one of those has an individual tree that you can draw. And we can count the nodes from one to the other. And we could say that one goes up 7 nodes and the other one has 6 nodes. Okay, we can do that.
I mean, you can put it that way, but that still seems kind of iffy.
It's not subjective. It's actual. It's real. This is how things develop. So why would you call that subjective when we know that it has to go through these stages to get all those tissue types?
Well, I mean, it just seems more reticulating of a process than the kind of descent-like pattern that you see when you do actual phylogenetic trees.
Oh, no, we're not talking about phylogenetic trees. We're talking about tissue trees. We're talking
I
about
know. That's what I mean, that you can't as easily draw a tree-like structure for embryological tissues as you can with genes or species.
No, you can. You can definitely draw these tree-like structures in embryology. Why would you not be able to?
Well, because I mean, like, one tissue isn't really descending from another. That's what the lines on a tree represent.
No, they actually are. They actually are descending from one another. So basically, you'll end up with a population of cells, like right here. Okay, and this is a neuron developing. Okay, and so you have a population of cells, and they're all the same. They're all type A cells, right? Okay, so one of these cells, because of noise, basically, one of these cells starts becoming more like it has a 0.01% advantage, basically. So it becomes a slightly different kind of cell. And when it becomes a different cell, it goes out and it feeds back to these cells and it tells them to lay down. Okay. So it's lateral inhibition is what it's called. So it laterally inhibits the cells around it. So the type B cells are these. So they're type B. Those become glial cells, and this becomes a neuron. That's how your neurons develop. So all cells are derived from this type A cell, Okay. And then they go to A prime, which would be a neuron, or they go to B, which would be a glial cell.
So
Okay.
you're talking like the property or quality of the cell is like being inherited or something.
Yes. Yeah, definitely.
Okay.
I'm
'Cause-
sorry,
'cause that-
I took over speed. I had to see Brainbug's puppy here.
Oh,
This
yes.
is Belle. She is 13 years old.
Oh, my
Oh.
God.
Wow.
The one you
Still a
hear
puppy.
yapping though in the background of my streams and stuff. She's the loudmouth,
Wow.
but she
Goodness
thinks she's
gracious.
protecting the house. It's cute.
Yeah, these cell types, they take on certain characteristics, and they have a proteome inside of them, basically a certain proteome. So they will, and all of these proteome basically have a certain signature, like there's certain genes that are being expressed, and then they will. So you have P1, basically proteome 1, and it will modify very slightly, and it will deride from that. You'll end up with P2 cells, and you have to go down these pathways in order to get that. That's how embryogenesis works. Are you convinced?
I guess for the moment.
Okay, good. We'll go further into that. Like for instance, we have a cell, right? And it's a certain shape, and it has a membrane.
Looks like a plant cell.
Yeah, it kind of does, but there's human cells that kind of look that way too. And in that membrane, there's basically, okay, we have the nucleus, and that nucleus is arranged in a certain fashion so that it is expressing a particular proteome. So there are certain proteins, RNAs, and whatever, I keep misspelling that are going to be created based on that cell, right? That type. And this cell has a couple of things operating on it. Like, for instance, it has transmembrane signal receptors, right? So it can feel its environment. And out here, we have, we have like an extracellular matrix, basically. And we also have a bunch of proteins out here that are outside of the cell, because some cells secrete, and there are basically gradients of these things that, on different sides of the cell, there might be less of these orange things, and this may have a lot of them. So there's that. So there's the environment that the cell lives in, okay? And that determines what the cell is going to do next, okay? And then there's other things, like if this cell divides and it ends up with another cell alongside of it, and it's in a small space, then these cells are going to put pressure on each other. So there's going to be a mechanical force on these cells, okay? And that mechanical force is also going to determine what the next cell does. And some cells will feed back positively to another cell and make that grow more quickly, and all sorts of weird things happen. So the point I want to make is that there's an environment of a cell that is both chemical and mechanical. And so there's a chemical and mechanical influence on every cell. And it will basically create itself to a certain point. Then it will influence other cells around it, and it will turn them into different cell types. So they used to be the same cell type. Now they're different cell types, that sort of thing.
Okay.
But it works in a tree, Andrew. It definitely goes in a tree-like fashion. Certain kinds of mesenchymal cells will, or certain kind of mesoderm will basically segregate into 3 types of mesoderm, and then with the middle type will segregate into something else. So it's very much a phylogeny when you look at an embryology. But,
Well, I mean, are they kind of, as these cells are developing, are they acquiring distinct, or I guess new, for lack of a better word, distinct characteristics that the the ancestral cells didn't possess?
Yes, yes. Based on what's coming in, like for signals, okay, mechanical and chemical signals. So we have two different kind of signals, okay? That will basically create a pathway down here that will end up influencing the genome, and it will start to express a different set of proteins, and it will become a different cell.
Or right, but the set of proteins was already there. Like, or I'm saying, like, in addition to, because when you're constructing a phylogeny, what you're basing it off of are these synapomorphies, and those are these uniquely shared traits. So I don't quite see how you can apply that here when you're just expressing different patterns of what's already there.
That's probably why they never talk about synapomorphies in embryogenesis. Because the cell has a particular proteome, okay, and then it gets modified, and then it starts creating proteome 2. So now it has different proteins, and it gets modified a little bit further, and it creates proteome 3, and then it happens to freeze there, so it no longer develops. But it starts out as cell type 1, turns into cell type 2, and then turns into cell type 3 based on its environment, based on where it is. Mechanical and chemical environment changes the cell type from one to another. So we're not talking, so if we think about it in terms of phylogeny, that's probably not a good idea, okay? Because it starts out.
That's why I'm saying, like, I don't think the phylogeny analogy doesn't work then.
I love cell cordoma.
Like you can say it's like tree-like, but I don't think you can say it's phylogeny-like.
No, I wouldn't. I'm not saying it's phylogeny-like. I'm saying it's tree-like.
Okay.
It's a network. It's a network. Okay. I guess if that's what you thought I was trying to say,
It,
no.
it should be reflected in phylogeny, though, right? I mean, we should see
Kind
these
of.
things change and adapt in the organisms that carry these traits, right?
Yeah, they kind of do evolve. You know, the cell evolves because it goes from cell type 1 to 2 to 3, and then it gets fixed at 3, right?
It has to be
That's
in the
what-
germ line, whatever, or the
And it's
what do you call it? The cells that reproduce.
we're not talking about
Has to-
reproduction. Huh?
Oh,
We're not.
I mean, if it has to save in those, I mean, to get to the next.
Yeah, if you end up
And-
with, like, say you have a tube of cells here, and I'm showing you a cross section of this tube of cells. The really cool thing is that this tube of cells will influence the cells on the inside to the point that they will start to die. Okay. And then you've got a hollow tubule, which becomes part of your vascular, you know, your blood system, your veins, arteries, or, you know, your lungs or whatever. So they'll start to influence the inside. So they started out as cell P1 type and ended up with P2 type, which is no cell at all or a dead cell. So these things will influence each other, and that's how these little tubes are formed in embryos. So how do we talk about that in terms of phylogeny? Well, that's a bad example for a phylogeny, but then sometimes you have like I was showing you in neurons. You know, you have this group of cells, and there's just a mess of cells, and one of them right here will suddenly take over and become a neuron, and all the other cells will diverge into glial cells. They'll wrap the neuron, and that happens over and over again. So they start out as cell P1, and they become either P2 or, you know, P3. So there's definitely a tree there. You start out at P1, and you become P2, which is a neuron, or P3, which is a glial cell. So that's a simple tree, right?
I guess so.
Yeah, they
Well,
started out as a
I think that Stupid Horror Energy had a question that I think that
Yeah, I was hoping I could share my screen so I could attempt to answer a question.
You were talking about synapomorphies earlier, and that links right into with what she's asking about. So
yeah.
Yeah.
Okay.
So she wants to show off her G-Man shirt?
No, it's what she said before that. If I could go up the phylogenetic tree, how is it that I can have one that is unrooted? And yeah, because we can. I'll let.
Oh, okay, good. You can go ahead and answer
I'll,
that.
I'll give my attempt at it, and then if I get something wrong, you can correct me.
Oh, you're fine. You probably know more about it than me.
Can I share my screen speed?
Yeah.
All right. So
Your screens are always longer than they are high.
yeah, it does seem to be the case, huh? So on the left here, we have an unrooted phylogeny, and what these kind of show is basically the close relationships between species. So in an unrooted phylogeny, there's no, like she said, up or down or higher or lower in the temporal sense, because you're just looking at the relationships between these various species. Whereas in a rooted phylogeny, where you have a definitive outgroup, basically that's the group that's most distantly related, or I guess that's less related than the group of interest you're looking at, I should say. In a rooted phylogeny, you root it at a particular node, and that determines the temporal axis. Basically, that determines how the traits evolve in such a way that okay, this right here is the common ancestor of this lineage and this lineage over here. So the basic difference is, in an unrooted phylogeny, you're just looking at close relationships between these terminal branches. In a rooted phylogeny, you're looking at the actual ancestry of what branch led to another branch through these branch points. Please let me know if that makes any sense at all.
Yeah, if you root the tree, by going to an outgroup, you're basically inferring a common ancestor, right? So
which we do with all of life. So you can root any tree all the way back to Luca, right? But you don't have to go back that far. You go back to a node.
Yeah. So once we decide what we think is the most likely root, then we're going to infer those node points in between, because we don't have them anymore. We've moved past that, right?
And that's how we build clades too, because a clade is the most animals that share the last common ancestor between these two, just like Dinosauria, for example, is the anything between that's closer related to a Triceratops and a barn sparrow falls into anything between those.
Basically, a clade is a common ancestor in all of its descendants. You can't leave any out. Or that's a monophyletic group, in other words.
Yeah. You can't be polyphyletic, because that doesn't work, and it's not good for. Well, you can be, because a lot of our colloquial names are like.
A lot of them are, but that's a no-no in cladistics.
Yeah.
Hey, we have an Ian Chen siding in the side chat. I just needed to mention that.
That's why we get such a language barrier with folks, because colloquial language, like words like monkey and fish, people have an idea what that is in their head, and it doesn't fit with our understanding of how life is changing on across the planet. It works fine in every day-to-day conversation, but you don't say a monkey, because to differentiate yourself from a monkey, say that's a monkey and I'm not, because by the law of monophyly, you would be a monkey if a monkey is everything that falls into the anthropoids or hominoids. Yeah. Sorry, I got off on a tangent. You go ahead. I steamrolled you again.
Yeah, there's script. That mutation rate isn't constant throughout evolution. It can change between different lineages, and it can change within a lineage. You can look at Dr. Dan's discussion, or I shouldn't say the discussion, but just his general focus on substitution rate versus pedigree rate, or mutation rate.
Yeah, over long periods of time, certain types of organism are, I mean, we can pretty much, they're kind of constant. It kind of averages out. But if you looked at a particular, like 3 generations, it's going to be all over the place, right? I mean, the mutation rates aren't going to be constant from generation to generation. Anyway,
Right. Yeah. The big thing is just that the mutation rate between different lineages isn't necessarily the same.
right. This is an example, Andrew, of a neuron being developed. Basically, you end up with these cells in the beginning. Okay, and the cell in the middle ends up. The cell in the middle starts out the same as the other 6 cells, or yeah, there are 6 cells. They're all the same, but one of them ends up through. They figured out in a recent paper, like 2022, that a small amount of noise one cell will suddenly get a little more of something than the other cells, and it will take over. And when it takes over, what it does is it laterally inhibits the cells around it, and it makes them have less and less of that protein. So the cell in the middle becomes a neuron, and the cells around become glial cells. So they start out as the same type of cell, and they differentiate into two completely different cells. And that's basically how your neurons develop, and a number of other tissues. But there are many other mechanisms for these things to happen, basically. But you're definitely looking at a tree where you start out with a primitive cell type, and then it differentiates into 2 final cell types. So,
sure, I can agree. It's tree-like in at least that sense.
Yeah, it tree-like. It's a tree.
Yeah.
So you can draw it as a tree. It's basically one cell type to another. I'm afraid you keep coming up with the idea that this is subjective. It is not. Basically, it is definitely a lineage.
Okay.
That's how organisms develop.
Well, I do have a limited understanding of how embryogenesis works. So
yeah. So, well, that's what I'm trying to talk about, and basically bring people to an understanding.
Well, anything that develops into different directions could be depicted with a tree, though, right? I mean,
yeah, you can.
Yeah, that's true.
Yeah,
we use human concept.
We use trees and networks for everything. I mean, biochemical pathways, and
you can do it with like languages, for example.
Yeah.
And there are things like that are just innate to us as growing up in Western society, I guess. Like when we look at a tree of language development, of evolution, of whatever, we know, that on the axis is that this is the passage of time, and these are the divergent points, and it's usually up or to the right that is the passage of time. But you were saying earlier about the unrooted trees. I just wanted to pop back over that real quick because I had a question about it, and we moved back into this other thing. So when you were talking about the rooted tree just being from a node of a common ancestor, would that be like what you were talking about previously to that with the synapomorphies? That's all I wanted to double back on.
So you're asking like how that's related to synapomorphies, or what synapomorphies is?
that you can distinguish that it's the last common ancestor. I generally understand that that's, and again I'll refer back to Dinosauria, like the hip placement and everything, and Dinosauria is the synapomorphies by which we differentiate them from other archosaurs.
Yeah, so the synapomorphy kind of defines a particular clade. What I think you're asking for is how we know that the outgroup is over here and the common ancestor is somewhere else. Is that kind of what you're asking?
Right. Yeah, and like does this converge over into their genetics? Does it roll over? Does that match when we look into these rooted trees? When we look at the last common ancestor, does it match genetically as well as with these synapomorphies that we've always traditionally built these clades on?
Well, yeah, synapomorphies though can be both morphological and molecular. You can have a particular site at a gene, and that particular nucleotide there, if it's different between the two, or it's shared between two lineages, that can be a synapomorphy as well. So it kind of, synapomorphy just means a character that is uniquely shared between two lineages with respect to a third one. So it can be morphological or DNA based or amino acids or whatever.
And that's how we nest the clades, is all these within this clade have this synapomorphy, and then these two groups both together have this other synapomorphy.
Right,
and
exactly.
so on and so forth. We nest them together like that. Excellent. Thank you
Yeah.
for diving into that just for a second with me.
yeah.
I like nerding out about this stuff.
Yeah, me too. Yeah, it's just to say one more thing. This is really why common descriptions of phylogenetics being based on similarity or things of that nature is kind of it kind of doesn't sit well with me, because you're not just looking at similarities per se. You're, like you were talking about, you're looking at these different characters with respect to different lineages. And I often like to say you're not just comparing apples with two lineages. You're comparing 3 lineages kind of at a time, at the very least. So that's kind of one misconception that I like to dig into.
Can trees connect?
Randolph, good to see you, by the way.
Hey, Randolph.
Hey, Randolph.
Hello,
Like
good to see
branches.
all of you as well.
I did want to quickly ask, Ian Chen seems to be having a debate. Does anyone know when that is at Aldal?
I have no
you
idea.
know the
Was it
date?
on?
Did I hear me
is it on
say
FFT's
May
channel?
5th, or was I muted? May 5th.
What's the debate about?
Evolution.
Oh, okay.
Ian Chen versus Kent Hovind.
Oh,
5th, you said?
May the 5th. Yep. 8
Okay,
p.m.
cool.
Eastern. So we have to convert it to real and actual time. But yeah, 7 p.m. Central.
it's always so interesting to see Ken Hovind's mountains of outdated evidence being used in these debates.
I know. So I predict that they're going to throw Ian out after 10 minutes.
They probably will. Let's hope not, though.
It's going to be the first time that basically Donnie threw out one of his debating. I don't know.
What is Ian's debate style? I'm not familiar with him.
It's humorous.
Yeah.
Oh, yeah, they won't like that.
Not gonna like that. Well, I think he's going to tone it down a little bit, but I'm not sure. But
Well, I just want to say good luck, Ian.
yeah,
Yeah, Ian, we're looking forward to it. I'll be there.
we'll see. Yeah.
Ian, I want you to have a lot of fun as well, even if
Yeah,
you can't
I mean, like, if
much
you're
about it.
going to debate Kent Hovind, you might as well have fun with it.
But did you see that statistic? So on SFT's channel, only 12% of the people there typically believe in evolution. So that's that's the lion's den. So
So back to my question earlier.
Well, 12%
It was-
believe in evolution? That's an interesting choice of phrasing.
yeah,
Can't.
well,
So,
I'd say most
am I
people
ready?
accept it as a fact.
yeah, I know.
Go ahead, Dot Dog.
Okay. So those trees, can they connect to each other? Like branches of science does?
Well, it's kind of just, they don't really connect to each other per se. They kind of just nest within one another. You just get a larger and larger tree depending on the group you're looking at.
Oh, okay.
So it's I mean, you could say, for example, I have a group over here, a tree, and a group over here, another tree, and you just connect them at a even more distant common ancestor, and you just get a bigger tree, if that makes sense.
Okay.
Yeah. So should we play this psi thing and see?
Sure.
I think we should.
Did you know
because-
that Gurr had the nerve to go live right in the middle of our
I
stream?
know. He's so rude.
What an asshole. Yeah. Okay. So let's see. Let me roll this thing. So you had so is that the right time stamp top dog 2406
34. Yep.
or 34? Okay.
Something.
Now, so do you think that the origin of life and biodiversity can be explained by natural processes? Like, so you said it's still a thing that we're working on and maybe we don't know, but like, are you confident that these can be explained by natural processes?
Well, I think everything can be explained by natural processes, which is consistent with belief in a creator God. I mean, nature is the creation of God, everything in it. Do I think that God had to come down and make a miracle or intervene specifically to make sure that, humans arose? Well, maybe yes. That's why I said I believe in that we're made in the image of God. How that happened, I don't know. I don't think we'll ever know.
You see, the thing is, like, when Cy started out there, he was doing okay, right? So he was like completely naturalism.
Yep.
God created nature. But then when it comes down to certain things like humans or eukaryotic cells or something like that, then he kind of backs up.
But
So-
later on, he'll say that God would make more sense that God is like making a process happen, not that
Okay.
he's individually making
he's
each
one of these-
animal.
So he's one of these process miracle people, sort of. Like God's fingers are constantly
Or
in it.
not like that. Oh, yeah, he
Oh.
wasn't thinking. He was thinking that he started the process, not that he's always in it.
Okay.
You'll see it later.
On the other hand, to say that God had to intervene continuously to create every new kind of species, that doesn't sound very divine to me. I mean, that doesn't sound like a very, like something that an all omnipotent, omniscient creator would do. Why wouldn't it make a lot more sense to devise a process, which we don't understand yet, but we're getting to devise a process that would create biodiversity? To me, that's more of a pointer to God than he said, okay, armadillos, let's see what's next. Pussycats, let's see what's next. One at a time, all several million species that have inhabited the planet. That may be what we would do, but God is not us. God is way beyond us, and just the creation of this universe is an amazing, thing. And the creation of life, which gets to your other question, because you asked about biodiversity. I don't have any problem with thinking that biodiversity can now be naturally explained. Origin of life is a different story. I don't think we will eventually explain it naturally. But I think when we do, we're gonna need some new ideas, because the ideas that we now have about how life could arise are not getting us anywhere.
Okay. And that's the Perry Marshall line right there. So Cy, and this is what I like about Cy, because he accepts like 99% of evolution, by natural means, because he believes the creator is smart enough to create a universe that's going to accomplish all of this automatically, right? Without divine intervention. But when it comes back to abiogenesis, he stops dead, and he insists that we're going to have to understand this new law of the universe that's going to have something to do with teleology. And that's where
we'll talk about that later.
He does. Okay.
Yep.
Yeah. That there has to be something teleological. Now, when I was talking about Lee Cronin with this assembly theory, and Lee Cronin is showing that teleology is basically also a natural process. That if you have a universe the way we do now, these teleological seeming assemblies will naturally arise. And once they do arise, they are teleological in the sense that they will do things like we do. Like we're the result of a naturally created teleological organism where we can plan for the future and all that stuff. We're kind of the extension of the way the universe works. And Perry Marshall and Cy would say we need a new law of physics for this, but Lee Cronin and Sarah Walker, I don't believe would say that. We have all the laws we need right now. It's just that complexity works this way. This is how it works. If you have these, if you have a periodic table of elements, you have a universe with the particles we have in it, you are naturally going to have life. You know, it's a natural result, that sort of thing. But it's we stumble over these terms. I think biologists like the term teleonomy, which I don't know for sure. Does anybody know the difference between teleonomy and teleology? I've looked at it a number of times, and I've never had them come right out and admit that teleonomy would be a teleological process or something that looks teleological, but it's perfectly natural in its formation, that it just naturally arises, that sort of thing. But anyway, I'd love to talk to Lee Cronin and Sarah Walker about this stuff, because that's kind of the crux of the problem, right? And Seigart will go 99 steps, you know, on that 100-step field, but he won't cross that line and say that God created a universe that basically is capable of creating humans, you know.
Hey guys.
So I have a Google definition of teleonomy if you want.
Yeah, what does it say?
Okay, I typed in difference between teleology and teleonomy, and the top of the line, there's a website called wikidiff.com. But anyway, it says, as nouns, the difference between teleology and teleonomy is that teleology is philosophy, in parentheses, the study of the purpose or design of natural occurrences, while teleonomy, and it has biology in parentheses, is the quality of living organisms of seeming to be organized towards the attainment of an end.
Okay, let me see if I can pop that up.
Hey guys, sorry to interject. I have to head out about now, but thanks for having me on.
Okay, Andrew.
Well, Andrew, thanks for stopping by. It's always a pleasure.
Oh, thanks. All right, see you guys.
Yeah, it was really enjoyable to have a disagreement with you over something.
I know.
Yeah.
I was thrilled.
Yeah.
Yeah, maybe we can do more sometime.
Yeah.
All right,
Hey,
guys.
thanks for coming by, Andrew.
Have a good one.
It's always a pleasure.
We'll explain why you're wrong later.
Yeah, go ahead. I probably am.
Okay. Teleology is philosophy, the study of purpose or design of natural occurrences. Okay. While teleonomy is a quality of living organisms seeming to organize towards the attainment of an end. Seeming to. Right.
In other words, it's a ID creationist kind of thing.
Yeah.
I don't know. Are there any philosophy of science people that are taken seriously who argue for teleonomy?
Teleonomy is no. Teleonomy takes the woo out of teleology.
Teleonomy is just the study of social organisms. It's not. Teleonomy is a ecological science. What you do when you're studying how like termites interact with each other, or how all the organisms in a lichen interact with each other and work for a mutual goal for the betterment of both organisms. I mean, there's different gradients of that. I mean, you got parasitism on one end, and then completely balanced mutualism, like how our cell has the bacteria absorb the bacteria. That's some perfect teleonomy there. So there's like a huge spectrum, but it's how organisms interact with each other, right?
Okay, teleonomy is the quality of apparent purposefulness or of goal-directedness of structures and functions in living organisms brought about by natural processes like natural selection. So teleonomy takes the woo out of teleology. Teleonomy takes the mind out of it. There's no mind. It's just natural processes do this, and they end up creating things that appear to be teleological. That's my understanding of it.
It's favorable for the two organisms to work together. So it's there's no mind behind it. When they work together, they have a better survival chance and better reproductive chance.
They wouldn't be alive if they didn't work together.
Right.
I put a link in the side chat to the Stanford Encyclopedia of Philosophy.
Oh, fuck.
They have an article on teleology and biology. Teleological notions in biology. Item number 2 is very interesting. It's called explanatory teleonaturalism.
Oh, cool.
So that sounds interesting.
Teleonaturalism.
Did you put it? You put it in regular
Yes,
chat?
in the side chat.
Okay, I'll see if I can throw it in
Yeah, teleological. Explanatory teleonaturalism, which is a phrase that only a philosophy website could come up with.
Yeah. Yep.
Let's continue on.
Gotta love it. Oh, so Gura's live right now. What a jerk.
He is live right now.
Yeah, so we should probably shoot her in the head here pretty quick.
Yeah.
Well, I thought this was a very thoughtful and engaging discussion of philology.
Yeah, I have more incentive now to basically discuss embryology, because apparently there are a lot of things that people don't know about it, and it's probably a worthwhile thing to start understanding, because I think when you look at evolution from that standpoint, when you see what's happening and how these new pathways are developed in that tree, then you start to understand how organs can actually be created or develop in evolution. You can see the organismal path, and I think it's really important. So I'm going to try to do that. Why did Top Dog and Randolph turn blue?
They're blue?
Yeah, on my StreamYard they turned blue for a second. I'm not sure.
Huh. Yeah, it happens on mine sometimes, but I don't know why.
Wow.
I thought maybe they heard some bad news and they're just,
Right. So
we have a question. We have a question quickly from Stupid Whore Energy. You guys ever heard of Mike, Gene, and front-loaded evolution?
No.
I thought that front-loaded evolution was.
Sorry, go ahead. Sorry.
I thought that front-loaded evolution was like the woo idea that all the inside your genes is there everything, but to make everything, and we all just kind of evolved towards specific ends. And we started with everything.
Okay, so Stupid Whore Energy, is that Miller and Behe? That's the first article that pops up. But what is Mike?
Is Mike Gene a real name, or is Gene possibly a cleverly chosen name for a character?
Yeah. Is that a real name?
That'd be an excellent name.
I have to wonder.
I think it actually is.
Top Dog did turn blue.
Hey.
You're right.
Yep.
Top Dog's really blue.
Yeah, I haven't turned blue. Have I? No.
No.
This is on purpose.
Just Top Dog.
The Design Matrix, a Conciliance of Clues by Mike Gene.
Okay, there's really a guy named Mike Gene. Oh, what did it, his mom? Okay, that's fake. Fake news. Fucking fake news. There's a geneticist named Mike Gene. Bullshit. I call bullshit on that.
That's amazing. He looks at microscopes all day and studies genes.
Well, maybe his mother was really controlling,
Yeah. She said, "You're going to go to school to be a geneticist."
Right.
Mike Gene.
Yeah.
Maybe he was just so into it that he changed his name to that. We don't know.
Could be. Well,
I'm not going to pry into the guy's personal life. These are his choices, what he wants to go by. But that's what it is on the nose. It's on the nose.
There's no description of this. There's no information about it. It's just a book.
I found
for $8.98.
I found a little podcast. I'll put it in the description. Mike Gene, What is Front Loading? In this chapter of Darwin or Design, I talk with Mike Gene. We talk about Mike's forthcoming book, The Design Matrix, and we discussed the idea of front loading in biology.
Yeah, that's kind of
Contributes regularly to the blog Telic Thoughts. Okay, there's a blog called Telic Thoughts. Don't ever have those, boys and girls.
Yeah.
Well, why not?
That way, doom-wise. Anything going on, Randolph, you want to talk about before I shoot her in the head?
Looks like we're going to have another one of those streams coming up on
Saturday?
on Sunday. Yeah, the fortnightly stream where people come in to try to convince anybody on the panel that a goddess or a god exists.
Has it been a fortnight already?
You mean
Yeah,
the
it's been a
weekly?
whole fortnight almost.
Okay, you mean that what he meant to say is the 2 weekly stream? Or the
Every
5?
2 weeks, yeah.
Yeah, because I call bullshit on this Fortnite stuff too. That's not a word.
Hey man,
That
look
is
at
what
the
a fortnight is.
You've never read Shakespeare then?
Not a word.
or Lord of the Rings.
Yeah, Lord of the Rings is where I learned. I can measure fortnights on that map.
Yeah, yeah, you remember that in that book.
Yeah.
So in that series, yeah, it's a lot of people think it's a trilogy, but it's actually 6 books. Lord of the Rings. So
Yeah, that's like Douglas Adams with his four book trilogy. The original joke. He took the joke.
yeah. So I actually on April 20th, I had 420 subscribers, and I ran a 420 stream at 4:20 p.m. with Neil, the 604 atheist, and he didn't smoke anything on the stream because of his medication. He can't. So he was totally focused, totally lucid, and it was great. It was very informative. I learned a lot about marijuana, and I've had some feedback from people who saw the stream and found it informative as well. The first one, unfortunately, had audio issues, but because I had speech to text kind of closed captioning going on, people could still understand what we're talking about at first. So the stream got cut off because of some issue with Google Meets, and so I started a second one. And that one went really well. And then we opened up the microphone, and Canadian Ollieman came on and showed us his collection of bongs and a hookah and some other things, pipes and whatnot. And it was so interesting. These guys were both wonderful. And then a lady from California came on, and we talked about legalization in California as well. Like Neil talked about legalization in Canada. So it's really, it's interesting to get the U.S. perspective in there as well. I enjoyed it. I haven't done many live streams. I haven't run many on my own, and this was a learning experience again, but it went out well, and I was quite happy to I'm happy to do that again. I'm thinking it's a series I decided I'm going to start called Fireside Chat, and it'll be one of the ones where we actually have a fire in the background. I had my fireplace going. It was a little hard to see. I got to work on that, but there was one time years ago here, a conservative political party, somehow I got invited to this thing. So I went with my friend, and it was called a Fireside Chat. Not a bit of fire anywhere. And I'm thinking, conservatives do stuff like this.
Well, wasn't that like from taking it from FDR? Didn't he do Fireside Chats? And then Jimmy Carter, I think, tried to do those as well during his administration. And so seems like conservatives will want to stay away from that idea.
Yeah, well, a fireside chat, you have some kind of a fireplace there and you're chatting around is what you better do, or some kind of fire around, just because it's a fireside chat. Otherwise, it seems
right. At least just hold out your lighter,
Yeah, something. Or an animation of it.
So Danny the Weenie is going to be on Leo's channel saying really dumb things about philosophy of the mind.
Or he could be saying smart things, Speed. Come on. Be charitable.
No.
What's a Walden pod?
Who the heck is it?
I don't know.
A what pot?
I don't know who Walden Pod is.
I don't know who Walden Pod is.
There's a question mark behind it, and
maybe nobody knows what Walden Pod is.
Is it a them or a what or who?
I've heard of Walden Pond.
So pod is Greek for foot, so it's Walden foot.
Walden foot. Oh. Yeah, I don't know.
Or maybe podcasts. Walden something podcast.
Maybe it was a
damn
typo.
It, Scott.
It could have been a typo. Okay, Bree, it could have been a typo.
Okay.
It could have been a typo. I'm just saying. I don't know. Could have been a typo.
Leo's usually pretty is in the margins though,
Yeah, I'm going to call that Leo and the Weenie in my description.
Leo and Das Weenie. Das Wiener.
Sometimes I
end up-
think that Speed of Sound is looking for a fight.
Yeah.
With Danny
Seems like it.
specifically.
Things have been nice and quiet.
What?
You not get enough excitement from all those raccoons?
What you saying? What you saying?
He's saying he wants Danny to talk about Jerry Fodor with him.
There you go,
Yeah.
top dog.
Yeah.
Oh, look at that fire. That's a beauty you got going there.
Oh, speaking of looking for a fight, I've been building a path down into the river valley, and I think it's about a 50, 60-foot drop, and it runs over, I don't know, several, maybe 500 feet. And so I've been running down there every day, cutting like with a chainsaw, and I have COPD, right? And I've been going down there with a chainsaw and cutting a path and with, clippers. And then every now and then I strap a 40-pound leaf blower on my back and I run down that hill and then come back up. And sooner or later, it's going to kill me. So I'm not going to be here very long.
Well, maybe you could throw like an oxygen tank on top of the leaf blower pack, and maybe that'll help you.
Come on, Speed of Sound. We want you to stick around. There's so much more science to cover.
And I'm going to be coyote food any day now. So please subscribe to my channel. You're going to feel bad if you don't.
Or raccoon rustlings.
Raccoon food, yeah. Anyway,
How many raccoons you got now?
at the most I counted 26. So
That's a lot.
Yeah, I've been cutting them down though because they're getting a little expensive to feed.
How are you
feeding
cutting
them
them
cat
down?
food?
Dog food.
Dog food?
I'm not buying them Iams. I'm giving them the cheap stuff from Sam's Club.
It's still better than what. It's still good for them.
Yeah.
How are you cutting them down?
Because I'm trying to. Well, the young ones. I'm afraid that the young ones coming up are going to become too dependent on me, and they're not going to know how to hunt for themselves. So I've cut them. I've I've been cutting them back every day, and I've got them down to about half what I was giving them.
Okay.
Have we ever mentioned what a group of raccoons is called?
Yeah, somebody came up with the name, and it's a weird name.
They're either called a nursery or a gaze, G-A-Z-E, gaze.
Yeah, gaze.
I think it was me a while back.
That would be a nursery.
A gaze.
Yeah.
A gaze
Nursery
of
of raccoons.
Wait a minute. People that gaze at nurseries are not good. Are not good things.
Unless you're the parent.
They arrest those people.
Yeah, you probably do want to cut the raccoons back because once you're coyote food, then they're going to start attacking all your neighbors looking for somebody to feed them.
Right. Yeah.
Swarms. Your neighbors are going to like one night in the middle of winter. There's just going to be swarms of raccoons all over their porches and windows.
You know, the raccoons have probably been studying you, and what they're going to do is do away with you, and one of them is going to impersonate you, and all the others are just going to hide behind the couch or whatever when the neighbors come to the door.
Maybe the
How do
weekend
we
that
know?
Bernie's them.
How do we know he's not a raccoon now?
So
No, we don't. We don't. He's got those bags under his eyes.
How are they going to pay the food bills? Oh, they're bandits. They'll steal it.
So for 20 years, I lived next to a virtually abandoned house, and 9 months ago a family moved in, and I was really worried about the raccoons. But the first time I fed the raccoons, the lady was out there. I think she was almost on her belly with a camera taking pictures of them. She was, "Oh, I love these things. We love it when you feed the raccoons."
Oh.
It like every night at 8:00 they get this little raccoon show. So anyway, I ended up getting the best neighbors ever. They're exactly like us, and they'd fit into this channel and everything. So they're pretty.
The ones with the kid who wants to do the bug stuff.
Right. Yeah. And yesterday he was accosting me and asking me where the hell that bug video is.
Yeah.
Yeah.
Where is it? We need to finish it before your raccoon food, probably.
We're gonna do it. I assure you. Okay, we got to get out of here, guys. Gur's live. We don't want to interfere with Gur.
Just wanted to say one thing quick about the front loading thing, DNA that those people say. Is it like somehow like something that they can't live without in genes? Is that what they meant, front loaded?
I think that maybe they're talking about created heterozygosity. I'm not sure. I think that's what they're talking about.
If they have in different species that are like similar to them, and then
Well, they're
if they
talking
say
about
this, like front loaded things, like whatever
Like-
it is they're talking about this, but it could be not in the other like species that are similar looking to them.
Well, they want to say that there were only two cats, two members of the cat kind that came off the ark, right? But they also want to say that 1,400 years prior, when God created the universe, that God created only two kinds of, or one kind of cat, but he front loaded the variation into the genome so that it could diversify into tigers and lynx and bobcats and all of that. So that's what I think they're saying, but I have to read it. I'll listen to the podcast when I linked the podcast in the description, so we can check it out and talk about it maybe tomorrow.
Okay.
Okay, boys and girls, we gotta go. I gotta kill this thing. How do I do that?
Well, stay curious.
Okay. Yep.
Bye, everyone. Thanks for tuning in.
Goodbye, cruel world.
Good talking with all of you.